The step lands before the realization forms. A foot presses into what appears to be nothing more than a rough patch of seabed, an irregular stone, a fragment of reef, and in that instant, the illusion fractures into a violent exchange of chemistry and pressure. Dorsal spines snap upward with mechanical precision, piercing skin and driving venom deep into tissue, while the victim's nervous system ignites in a cascade of signals so intense that coordination collapses almost immediately. What seemed inert reveals itself as a device primed for the contact, a biological mine engineered not to pursue, but to punish the slightest intrusion with overwhelming force. The stonefish does not stalk, and it does not chase. It occupies. Its body is sculpted by evolutionary pressures into a near-perfect mimic of its surroundings, a living extension of rock and sediment that dissolves into the visual noise of the ocean floor. Irregular contours, mottled pigmentation, and textured skin break up its outline, exploiting the limitations of predator and prey perception systems that rely on pattern recognition and edge detection. To the passing eye, it is not a fish at all, but a discontinuity in the substrate, a harmless anomaly in a landscape of fragments. This is not simply camouflage. It is perceptual manipulation, a strategy that turns the environment itself into a shield. Yet the true power of the stonefish lies not in its invisibility, but in the system it deploys when that invisibility is breached. Along its back, a series of stout, needle-like spines are connected to venom glands capable of delivering a complex biochemical payload. Each spine functions as both a sensor and a conduit, responding to pressure with immediate mechanical and chemical action. When compressed, the spines drive downward into the offending tissue, and the glands force venom through channels within the spines, injecting it directly into the wound. This is not a gradual process. It is instantaneous, converting physical contact into a rapid infusion of toxins. The venom itself is a sophisticated mixture designed to disrupt multiple physiological systems simultaneously. Components within the toxin interfere with cell membranes, causing localized destruction of tissue that manifests as swelling, necrosis, and intense pain. Other elements affect cardiovascular function, destabilizing blood pressure, and so on, in severe cases, leading to systemic shock. The pain response is not incidental. It is a critical part of the weapon's design, triggering a cascade of stress responses that further compromise the victim's ability to function. In this way, the stonefish's venom operates on both structural and neurological levels, dismantling the body's integrity while overwhelming its control systems. This dual-layered attack reflects a fundamental constraint of the stonefish's ecology. Unlike active predators that can pursue and subdue prey, the stonefish relies entirely on the moment of contact. There is no second chance, no extended struggle. The interaction must be decisive, delivering enough force and chemical disruption to neutralize the threat or deter the predator immediately. The venom, therefore, is not optimized for efficiency over time, but for maximum impact in a single, brief encounter. It is a solution shaped by stillness, where the absence of movement demands the presence of overwhelming potency. The apparent passivity of the stonefish conceals a deeper form of dominance. By embedding itself within the substrate, it becomes a fixed point around which other organisms must navigate. Small fish, crustaceans, and invertebrates move through the environment, unaware that they are passing within striking distance of a predator that does not need to reveal itself. When prey ventures too close, the stonefish can execute a rapid suction feeding mechanism, expanding its mouth cavity to create a vacuum that draws the organism inward in a fraction of a second. Here, the narrative shifts subtly. The same stillness that defines its defensive strategy also underpins its method of feeding. The mind does not only punish, it also captures. This convergence of defense and predation introduces a narrative reversal that challenges the initial perception of the stonefish as purely reactive. Its immobility is not a limitation, but a unifying principle that governs multiple aspects of its biology. The same morphological adaptations that allow it to avoid detection also position it optimally for ambush. The same venom that deters predators also ensures that any accidental contact becomes a lethal opportunity. In this way, the stonefish collapses the distinction between offense and defense, operating within a narrow band of behavior that maximizes efficiency across both domains. The energy economy of this strategy is particularly striking. Movement is metabolically expensive, especially in environments where resources can be patchy and competition intense. By minimizing locomotion, the stonefish reduces its energy expenditure to a baseline level, investing instead in the maintenance of its venom apparatus and camouflage. This trade-off reflects a broader principle in evolutionary biology, where organisms allocate resources toward traits that provide the greatest return under specific environmental conditions. For the stonefish, the return on stillness is amplified by the lethality of its chemical arsenal, creating a system where minimal input yields maximal outcome. However, this specialization comes with its own vulnerabilities. The reliance on camouflage and immobility means that the stonefish is highly dependent on the stability of its environment. Changes in substrate composition, water clarity, or the behavior of surrounding organisms can affect its ability to remain undetected. Moreover, certain predators have evolved strategies to circumvent its defenses, either by avoiding the substrate where it resides or by possessing resistance to its venom. This introduces another layer of complexity, where the stonefish's dominance is not absolute but contingent on a dynamic interplay of ecological factors. Within the broader ecosystem, the stonefish exerts an influence that extends beyond individual interactions. Its presence creates zones of risk that shape the movement patterns of other species. Fish may alter their foraging routes, invertebrates may adjust their burrowing behavior, and even larger predators may exhibit caution in areas where stonefish are known to occur. These behavioral modifications can cascade through the food web, affecting predator-prey relationships and the distribution of energy within the system. The stonefish, by virtue of its chemical and physical defenses, becomes an anchor point in the ecological network, a silent regulator of movement and interaction. There is also an underappreciated biochemical dimension to this influence. The compounds within the stonefish's venom are not static. They are subject to variation and refinement over evolutionary time. Different populations may exhibit variations in venom composition, reflecting local selective pressures, such as the types of predators encountered or the nature of the prey available. This chemical diversity contributes to the adaptability of the species, allowing it to maintain its effectiveness across a range of environments. In this sense, the stonefish's venom is not just a weapon, but a dynamic system, continuously shaped by the ecological context in which it operates. The interaction between the stonefish and its environment can also be viewed through the lens of information flow. Camouflage disrupts the transmission of visual information, preventing other organisms from accurately identifying the stonefish as a threat. The venom, in turn, serves as a form of chemical communication, delivering a clear and immediate signal when the camouflage fails. Together, these elements create a layered system of interaction, where the absence of information is followed by an overwhelming presence of it, encoded in pain and physiological disruption. This sequence, concealment followed by revelation, defines the stonefish's mode of existence. As the initial shock of contact subsides, the consequences continue to unfold within the victim's body. Tissue damage progresses, inflammatory responses intensify, and systemic effects may develop depending on the severity of the envenomation. For smaller organisms, the outcome is often fatal, their bodies becoming a source of nourishment for the stonefish. For larger animals, including humans, the experience can be debilitating, a stark reminder of the power contained within an organism that rarely moves and is almost never seen. In the end, the stonefish embodies a form of power that is easy to overlook. It does not announce itself, it does not display, and it does not pursue. It simply exists within the environment, perfectly aligned with its surroundings, waiting for the moment when contact transforms invisibility into impact. Its strategy is not built on speed or strength, but on the precise integration of form, chemistry, and behavior. The perfect ambush mine is not a metaphor, but a literal description of a system where the boundary between organism and environment dissolves, and where the cost of a single misstep is measured in the rapid unraveling of biological function. The contact happens before the thought. A translucent bell drifts through warm coastal water, almost invisible, and then, without warning, a filament unfurls, touches flesh, and an electrical storm erupts across living tissue. In less than a fraction of a heartbeat, thousands of microscopic harpoons fire in perfect synchrony, each one penetrating skin, anchoring, and injecting a molecular payload designed not to wound, but to shut down. What appears to be a passive encounter is, in reality, one of the fastest and most coordinated biological attacks on Earth. A system so refined that the victim's nervous system begins to fail before the brain can even register pain. The box jellyfish operates as a decentralized weapons platform, its body composed mostly of water, yet armed with an array of nematocysts, microscopic capsules that function as single-use biochemical syringes. Each nematocyst contains a coiled tubule under extreme internal pressure, primed to fire when triggered by the slightest mechanical or chemical cue. The release is not gradual, it is explosive, driven by osmotic forces that accelerate the tubule outward at speeds that rival mechanical projectiles at the microscale. This is not a sting in the conventional sense. It is a coordinated barrage, a simultaneous discharge of thousands of independent injectors that collectively overwhelm the target's physiological systems. Once embedded, the venom begins its work with ruthless efficiency. It is not a single compound, but a complex cocktail of proteins and peptides, each targeting a different system within the body. Some components disrupt ion channels in nerve cells, preventing the transmission of electrical signals and effectively silencing communication between brain and muscle. Others attack cardiac tissue directly, destabilizing the rhythmic contractions that sustain life. Still others break down cellular membranes, causing localized tissue destruction that amplifies the overall effect. The result is not merely pain, although the sensation is often described as excruciating, but a cascading failure of multiple biological systems occurring in parallel. In the open ocean, where space is vast and escape routes are plentiful, Speed is everything. Prey cannot be chased. It must be neutralized instantly. The box jellyfish's strategy reflects this constraint. Rather than investing in muscular strength or complex pursuit behaviors, it has evolved a system that converts contact into immediate control. The moment a fish brushes against its tentacles, the outcome is already determined. Paralysis spreads through the prey's body in seconds, immobilizing it before it can flee. The jellyfish then draws the incapacitated organism toward its central mouth, completing a process that began with a single, almost imperceptible touch. Yet the true sophistication of this system lies not only in its speed, but in its selectivity. Nematocysts are not triggered indiscriminately. They require specific combinations of mechanical pressure and chemical signals to fire, reducing the likelihood of wasted discharge. This is critical because each nematocyst can be used only once. After firing, it must be replaced through cellular regeneration, a process that consumes energy and time. The jellyfish, therefore, operates under a constraint that shapes its entire biology. Every shot must count. This limitation has driven the evolution of a triggering mechanism that balances sensitivity with precision, ensuring that the weapon is deployed only when success is highly probable. The box jellyfish's body plan further enhances its effectiveness. Unlike many of its gelatinous relatives, it possesses a surprisingly advanced sensory system. Clusters of simple eyes, known as rhopalia, are embedded along the edge of its bell, allowing it to detect light, obstacles, and even the silhouettes of potential prey. This visual capability enables it to navigate complex environments such as mangrove forests and shallow coastal waters, where obstacles are abundant and prey density is high. The jellyfish is not drifting blindly. It is actively positioning itself within the environment, aligning its tentacles with the most likely paths of passing organisms. This introduces a subtle but profound shift in understanding. The box jellyfish is often perceived as a passive drifter, a creature at the mercy of currents. In reality, it is an active participant in its own predatory strategy, using sensory input to optimize the placement of its chemical weapons. The apparent fragility of its gelatinous form conceals a system of remarkable integration, where movement, perception, and biochemical attack are tightly coupled. The weakness of lacking a rigid body or powerful muscles is not a limitation, but a specialization, allowing it to invest its energy into the development of one of the most efficient venom delivery systems in the natural world. The ecological implications of this strategy extend beyond individual encounters. By targeting small fish and planktonic organisms, the box jellyfish influences the structure of local food webs. Its ability to rapidly immobilize prey reduces the likelihood of escape and increases predation efficiency, which can alter population dynamics within its habitat. In regions where box jellyfish are abundant, their presence can shape the behavior of other species, driving them to avoid certain areas or depths. This, in turn, affects patterns of feeding, reproduction, and migration, creating a cascade of effects that ripple through the ecosystem. There is also a deeper, more intricate layer to this chemical warfare, one that involves symbiosis at the microscopic level. The venom itself is not solely the product of the jellyfish's own cells. Evidence suggests that associated microorganisms may play a role in the synthesis or modulation of its toxic compounds. This means that the box jellyfish is not acting alone, but as part of a complex biological network where multiple organisms contribute to the creation of its lethal arsenal. The weapon is not just an adaptation of a single species, but a collaborative outcome of evolutionary interactions across different domains of life. And yet, for all its lethality, the box jellyfish is not invincible. Certain species have developed resistance to its venom, exploiting ecological niches where others cannot survive. Some fish, for example, possess protective mucus layers or physiological adaptations that reduce the effectiveness of the toxins, allowing them to navigate tentacle-laden waters with relative safety. This introduces a narrative reversal that challenges the initial perception of absolute dominance. The very system that grants the jellyfish its power also creates selective pressure for resistance, setting the stage for an ongoing evolutionary arms race. In this context, the box jellyfish's venom is not merely a weapon, but a driving force in the evolution of other species. It shapes behaviors, influences physiological adaptations, and contributes to the diversification of life within its environment. What begins as a localized interaction between predator and prey expands into a broader dynamic that affects entire communities. The instant shutdown system, as efficient as it is, becomes one component of a larger, ever-shifting balance. At its core, the box jellyfish represents a fundamental principle of life in the ocean, that dominance is not achieved through brute force, but through precision, efficiency, and integration. Its body, almost entirely composed of water, houses a system capable of delivering one of the most rapid and effective biological attacks known. It does not chase, it does not struggle, and it does not hesitate. It simply waits, senses, and executes, transforming a fleeting moment of contact into a decisive outcome. The encounter ends as quickly as it began. The tentacles retract, the prey is drawn inward, and the surrounding water returns to its deceptive calm. There is no visible trace of the violence that has just occurred, no lingering disturbance to mark the event. But within that brief interaction lies a convergence of physics, chemistry, and biology so finely tuned that it blurs the line between organism and machine. The box jellyfish does not merely survive in the ocean, it operates within it as a system of instantaneous control, where the difference between life and death is measured not in seconds, but in the time it takes for a microscopic harpoon to fire. The reef fractures, not with noise, but with absence. A juvenile grouper approaches, posture confident, its trajectory calculated by instinct and experience, and then it hesitates. No strike, no chase, just a sudden, inexplicable withdrawal as a striped silhouette drifts forward, slow, deliberate, and untouched. The lionfish does not accelerate, does not pursue, and yet the space around it clears as if pushed back by an invisible force field. This is not fear born from encounter. It is avoidance, encoded into the nervous systems of those that have learned too late. The predator has arrived, but it does not need to announce itself with violence. Its power lies in the quiet certainty that nothing here is equipped to challenge it. The lionfish carries its weapon openly, an array of elongated dorsal, pelvic, and anal spines that appear ornamental but function as a distributed injection system. Each spine is connected to venom glands that produce a complex mixture of proteins designed not for immediate lethality, but for intense pain, neuromuscular disruption, and physiological shock. When pressure is applied, the thin sheath covering each spine is pushed downward, exposing grooves through which venom flows directly into the tissue of the attacker. Unlike ambush predators that rely on surprise and rapid incapacitation, the lionfish deploys its chemical arsenal as a deterrent. a biological warning system that transforms attempted predation into a costly mistake. This distinction is critical. The venom of the lionfish is not optimized to kill instantly, but to discourage interaction, to create a persistent memory of pain that alters behavior long after the encounter. Predators that survive a lionfish sting often avoid similar shapes and patterns in the future, effectively extending the reach of the lionfish's defense beyond the individual. This is chemical warfare operating through learning and memory, a strategy that reduces the need for repeated confrontation by shaping the decision-making processes of other species. The result is a predator that moves through its environment with an unusual degree of freedom, unburdened by the constant threat of being hunted. In its native Indo-Pacific range, this system functions within a balanced network of interactions. Predators, such as large groupers and sharks, may still consume lionfish, either through learned techniques that avoid the spines or through physiological tolerance to the venom. Here, the lionfish is one participant among many, its chemical defenses integrated into a broader ecological context where checks and balances maintain equilibrium. But when this species is introduced into new environments, the rules change dramatically. In the Western Atlantic and Caribbean, the lionfish encounters ecosystems that have not co-evolved with its presence. Native predators lack both the behavioral strategies and the physiological adaptations required to handle its venom. The warning encoded in its spines is not recognized, and the cost of learning is often too high. As a result, the lionfish experiences a release from predation pressure, a condition that allows its population to expand rapidly. This is where the narrative shifts from individual survival to ecological transformation. The same venom that once served as a defensive measure becomes a tool for territorial expansion, enabling the lionfish to occupy niches that would otherwise be contested. The feeding behavior of the lionfish amplifies this advantage. Unlike many predators that rely on speed or endurance, the lionfish uses a methodical approach, herding small fish into confined spaces using its broad, fan-like pectoral fins. These fins act as barriers, reducing the escape options of the prey and guiding them toward the lionfish's mouth. Once within range, the lionfish executes a rapid suction feeding mechanism, expanding its buccal cavity to generate a vacuum that pulls the prey inward. This combination of slow approach and sudden capture minimizes energy expenditure while maximizing success, a strategy well suited to environments where prey density can fluctuate. The absence of effective predators allows this feeding strategy to operate unchecked. Juvenile fish, including those of ecologically and economically important species, become primary targets. As lionfish populations grow, their cumulative impact on these prey communities intensifies, leading to significant reductions in recruitment and altering the structure of the reef ecosystem. This is not a gradual shift, but a cascading effect where the removal of key species disrupts the balance of competition, predation, and resource availability. The lionfish, through its chemical deterrence and efficient feeding, becomes an agent of ecological change. Yet the story does not end with dominance. As lionfish populations expand, they begin to encounter the limits of their own success. Increased density leads to competition among lionfish themselves, driving changes in behavior and distribution. Some individuals move into deeper waters or less optimal habitats, extending the range of the invasion, but also exposing themselves to new challenges. In certain regions, native predators have begun to adapt, learning to consume lionfish by targeting them in ways that minimize the risk of envenomation. This represents a subtle but important reversal, where the initial advantage conferred by chemical defense begins to erode as the ecosystem responds. There is also a human dimension to this dynamic. Divers and fishermen, recognizing the impact of lionfish on reef systems, have initiated removal efforts that introduce a new form of predation. The venom that deters natural predators does not prevent humans from developing tools and techniques to capture and consume lionfish, effectively reintroducing a control mechanism into the system. In some areas, this has led to localized reductions in lionfish populations, demonstrating that even a highly effective chemical defense can be circumvented by innovation and coordination. At a physiological level, the lionfish's venom reflects a balance between potency and sustainability. Producing venom is metabolically costly, requiring the synthesis of complex proteins and the maintenance of specialized glandular structures. The lionfish mitigates this cost by using its venom sparingly, deploying it primarily in defensive situations, rather than as a primary means of subduing prey. This contrasts with species that rely on venom for hunting, where frequent use necessitates rapid replenishment. In the lionfish, the venom functions as a long-term investment, a protective measure that enables other aspects of its biology to operate more effectively. The visual display of the lionfish further enhances this system. Its bold stripes and elongated fins create a distinctive silhouette that is easily recognized, even in the complex visual environment of a coral reef. This aposematic coloration serves as a warning signal, reinforcing the association between appearance and danger. Over time, this reduces the likelihood of physical contact, conserving the lionfish's venom reserves and minimizing the risk of injury. The combination of visual and chemical defenses creates a multi-layered deterrent, one that operates both before and after contact. The ecological role of the lionfish, particularly in invaded regions, illustrates the broader concept of how chemical defenses can influence not just individual interactions, but entire ecosystems. By reducing predation pressure and enabling population growth, the lionfish alters the flow of energy through the food web. Prey species decline, competitors are displaced, and the overall diversity of the reef can be affected. This is not merely a case of one species outcompeting another, but a reconfiguration of ecological relationships driven by a single, highly effective adaptation. In this context, the lionfish embodies a form of expansion that is both biological and chemical. Its venom does not need to be deployed frequently to exert its influence. Its mere presence, combined with the memory of pain it creates, is sufficient to reshape behavior and interactions. The reef does not resist with force, but adapts slowly and unevenly to the new conditions imposed by this invasive predator. The process is ongoing, a dynamic interplay between the lionfish's chemical defenses and the evolving responses of the ecosystem. As the lionfish drifts through the water column, its movements remain unhurried, almost indifferent to the changes it is driving. The spines that line its body continue to signal danger, the stripes continue to advertise its presence, and the fish around it continue to adjust their behavior in response. What began as a defensive adaptation has become a mechanism for expansion, a tool that allows the lionfish to extend its reach across vast stretches of ocean. The expanding chemical empire is not built through conquest in the traditional sense, but through the quiet, persistent reshaping of ecological boundaries, one avoided encounter at a time. The bite closes before the mistake is understood. A reef predator lunges, jaws compressing around what appears to be a slow, defenseless fish, and in that instant, the exchange is sealed. not as a struggle of strength, but as a transfer of chemistry that begins dissolving control from the inside out. Muscles that once powered pursuit seize into silence, electrical signals fail to propagate, and the hunter's body, built for dominance, becomes an inert vessel, drifting in open water. The pufferfish does not resist, does not counterattack, and yet it has already won. Its weapon is not deployed in the moment of conflict. It was present once before the encounter began, embedded within its tissues as a constant, invisible threat. The pufferfish embodies a radical departure from conventional, predatory, or defensive strategies. It does not invest heavily in speed, armor, or aggressive retaliation. Instead, it carries within its body a compound so potent that it transforms every interaction into a high-stakes gamble for any organism that attempts to consume it. Tetrodotoxin, the molecule at the center of this strategy, is a neurotoxin that targets voltage-gated sodium channels, the microscopic gates through which electrical signals travel along nerve and muscle cells. By binding to these channels, the toxin prevents the flow of sodium ions, effectively halting the transmission of electrical impulses. Without these impulses, muscles cannot contract, nerves cannot communicate, and the coordinated activity required for life collapses into paralysis. This is not a gradual process. The disruption of sodium channels occurs rapidly, and its effects cascade through the body with precision. The predator that ingests a pufferfish may initially show no signs of distress, continuing its movements as if nothing has changed. Then, almost imperceptibly, coordination begins to fail. Swimming becomes erratic, responses slow, and within moments, the organism loses control of its musculature entirely. Respiration, driven by rhythmic muscle contractions, ceases, and the predator, once an active force within the ecosystem, becomes a passive object subject to currents and gravity. The pufferfish, by contrast, remains intact, its body carrying the same chemical warning that will deter future attempts. What makes this system particularly remarkable is that the pufferfish does not synthesize tetrodotoxin itself. The toxin is produced by symbiotic bacteria that reside within the fish's tissues, particularly in organs such as the liver, skin, and gonads. This relationship transforms the pufferfish into a host for a microbial defense network, a living reservoir of poison that is maintained and replenished through biological collaboration. The fish provides habitat and nutrients, while the bacteria produce the compound that ensures the host's survival. This is not merely an adaptation of a single species, but a partnership that spans different domains of life, integrating microbial metabolism into the macroscopic behavior of the fish. The distribution of tetrodotoxin within the pufferfish's body is not uniform, but strategically concentrated in areas most likely to be targeted by predators. The skin, which forms the first point of contact, contains significant levels of the toxin, ensuring that even a superficial bite can deliver a potent dose. Internal organs, which would be consumed if the predator persists, contain even higher concentrations, creating a layered defense system that escalates with the depth of the attack. This arrangement reflects a principle of deterrence rather than combat. The goal is not to engage and overcome an attacker, but to impose such a severe cost that the behavior is eliminated from the predator's repertoire. The inflation mechanism, often associated with the pufferfish, plays a secondary but complementary role in this strategy. When threatened, the fish rapidly ingests water or air, expanding its body into a spherical shape that is more difficult to swallow. This physical transformation increases the apparent size of the fish, complicating the mechanics of predation for gape-limited predators. Spines that are otherwise flattened become erect, adding a further layer of difficulty. However, this display is not the primary defense. It is a visual and mechanical distraction, buying time and increasing the likelihood that the predator will reconsider before committing to a bite. The true deterrent remains the chemical payload within. This combination of physical and chemical defenses creates a system where the pufferfish rarely needs to engage in direct conflict. Predators that have experienced the consequences of tetrodotoxin often learn to avoid similar shapes and behaviors, embedding the lesson within their neural circuits. Over time, this learned avoidance can extend beyond individual predators to influence population-level behavior, reducing the frequency of attacks on pufferfish altogether. In this way, the toxin operates not only as a direct defense, but as a driver of behavioral change within the ecosystem, shaping interactions through the memory of past encounters. Yet this apparent invulnerability is not absolute. Certain predators have evolved the ability to tolerate or even exploit the presence of tetrodotoxin. Some species of snakes and fish possess modified sodium channels that are less susceptible to the toxin's binding, allowing them to consume pufferfish without experiencing the same level of paralysis. This introduces a critical narrative reversal, where the ultimate deterrent becomes a selective pressure that drives the evolution of resistance. The pufferfish, once a near untouchable entity, becomes a specialized resource for those capable of circumventing its defenses. The chemical weapon that ensures its survival also creates a niche for organisms that can overcome it, illustrating the dynamic interplay between defense and adaptation. Within the broader ecological context, the presence of pufferfish influences the distribution and behavior of other species. Areas with high densities of toxic prey may see reduced predation pressure on other organisms as predators adjust their foraging strategies to avoid the risk associated with tetrodotoxin. This can lead to shifts in community structure, where the indirect effects of the pufferfish's defense ripple through the food web. The toxin, while contained within individual bodies, exerts an influence that extends far beyond, altering the flow of energy and the balance of interactions within the ecosystem. The metabolic cost of maintaining this defense system is not insignificant. Hosting toxin-producing bacteria, transporting and storing the compound, and ensuring that it remains effective all require energy and physiological regulation. However, the return on this investment is substantial. By reducing the need for constant vigilance and rapid escape responses, the pufferfish can allocate energy toward growth, reproduction, and other aspects of its life history. This trade-off highlights a fundamental principle of evolutionary strategy: the optimization of resource allocation in response to environmental pressures. For the pufferfish, the investment in chemical defense yields a level of security that few other organisms can achieve. At the molecular level, tetrodotoxin represents a precise and targeted intervention in the fundamental processes of life. By interfering with sodium channels, it disrupts the very mechanism that allows cells to communicate and coordinate. This specificity is what makes the toxin so effective, as it targets a system that is conserved across a wide range of organisms. The universality of this mechanism ensures that the toxin has broad applicability as a defense, capable of affecting multiple potential predators with equal efficiency. In this sense, the pufferfish's strategy is not tailored to a single threat, but to the underlying biology of its environment. The visual presence of the pufferfish, often characterized by its distinctive shape and patterns, reinforces this chemical defense. Bright coloration, in some species, serves as an aposematic signal, warning potential predators of the danger associated with consumption. Even in species that lack vivid colors, the unique morphology of the pufferfish can become associated with toxicity through learned experience. This integration of visual and chemical signals creates a multi-layered defense system that operates both before and after contact, reducing the likelihood of attack and ensuring that any attempt carries significant consequences. As the predator drifts, its body unresponsive, the pufferfish resumes its slow, deliberate movement through the water. There is no chase, no visible victory, only the quiet continuation of a strategy that relies on being left alone. The ocean around it remains active, filled with interactions and pursuits, yet the pufferfish occupies a different space within this dynamic. It is not defined by what it does to others in the moment, but by what it represents: a persistent, invisible risk that reshapes behavior and decision-making across the ecosystem. In this way, the pufferfish stands as a testament to the power of chemical deterrence. It does not need to demonstrate its strength repeatedly, nor does it need to engage in frequent conflict. Its dominance is established through the potential of its toxin, a constant presence that transforms every encounter into a calculation of risk. The nuclear option is not a weapon that is deployed lightly. It is a condition of existence, a state in which the cost of aggression is so high that it becomes, for most, unthinkable. The strike is already over before the water can react. A slender body flickers through a cloud of baitfish, and in that compressed instant, a single fish convulses, not from impact, but from something far more insidious, a molecular interruption that spreads faster than any physical wound. There is no prolonged struggle, no chase unfolding across open water, only a brief contact followed by a silence where movement should be. The fish's muscles cease to obey, its propulsion collapses, and gravity, so often irrelevant in the buoyant ocean, begins to reclaim it as it sinks, alive, but already functionally absent. The sea snake does not accelerate to follow. It slows, circles, and waits, because the outcome has already been decided at the level of chemistry. The sea snake represents a convergence of evolutionary pressures that have refined venom into a system of extraordinary efficiency. Unlike many terrestrial snakes that must contend with varied prey types and environmental constraints, sea snakes operate within a medium where escape is multidirectional and immediate. Water removes frictional limitations, allowing prey to dart in any direction with minimal resistance. To counter this, the sea snake's venom is not merely potent, it is specialized to collapse muscular function with remarkable speed. The primary components are neurotoxins and myotoxins, molecules that target the very mechanisms that enable movement. By binding to receptors at the neuromuscular junction, these toxins prevent the transmission of signals from nerve to muscle, effectively severing the connection between intent and action. This disruption is precise. The toxins do not indiscriminately destroy tissue. They interfere with communication, halting the cascade of electrical impulses that drive contraction. At the same time, myotoxic components begin to degrade muscle fibers, ensuring that even if partial signaling remains, the structural integrity required for movement is compromised. The result is a rapid onset of paralysis that renders the prey incapable of swimming, breathing, or escaping. In an aquatic environment, where respiration is often tied to continuous movement or active ventilation, paralysis is equivalent to suffocation. The prey does not die from external trauma, but from the cessation of its own biological processes. What makes this system particularly striking is its efficiency. Sea snakes produce venom that is among the most potent of any reptile, yet they deploy it with restraint. Unlike species that rely on repeated strikes or prolonged envenomation, sea snakes often deliver a single, controlled bite, injecting just enough toxin to achieve the desired effect. This is chemical warfare, calibrated to minimize waste. Venom production is metabolically expensive, requiring the synthesis of complex proteins and the maintenance of specialized glands. By optimizing the dosage and delivery, sea snakes ensure that each use of venom yields maximum return with minimal expenditure. The morphology of the sea snake supports this strategy. Its elongated, laterally compressed body allows for agile movement through water, while its paddle-like tail provides propulsion that is both efficient and precise. The head is streamlined, reducing drag and enabling rapid, targeted strikes. Unlike many land snakes, which may rely on constriction or prolonged engagement, the sea snake's anatomy is adapted for brief, decisive interactions. It approaches, strikes, and withdraws, allowing the venom to complete the process. This separation of action and outcome is a defining feature of its hunting strategy, where the physical encounter is only the initiation of a process that unfolds within the prey's body. The sensory systems of the sea snake further enhance its effectiveness. Vision plays a role, but chemical detection is equally important, allowing the snake to track the subtle cues released by its prey. Once envenomated, the immobilized fish may emit signals associated with stress or injury, guiding the snake back to its target. This creates a feedback loop where the initial strike is followed by a period of tracking and retrieval, ensuring that the energy invested in the attack is not lost. The snake does not need to maintain contact. It only needs to locate the result of its chemical intervention. At first glance, the potency of sea snake venom suggests a creature that poses a significant threat to any organism it encounters, including humans. Yet the reality presents a notable reversal. Sea snakes are generally reluctant to bite humans, and when they do, they often deliver little to no venom. This behavior reflects a fundamental principle of their biology. Their venom is not a weapon for indiscriminate use, but a specialized tool for capturing prey. Humans do not fit into this ecological role, and the snake's sensory systems likely recognize this mismatch. Engaging with a large, non-prey organism carries risks that outweigh any potential benefit, leading to a strategy of avoidance rather than confrontation. This restraint is not merely behavioral, but also physiological. Sea snakes can control the amount of venom they inject, a capability known as venom metering. By adjusting the dosage based on the context of the interaction, they conserve their resources and reduce unnecessary expenditure. In encounters with humans, this often results in dry bites, where no venom is delivered. This is not an act of mercy, but a reflection of optimization. The snake's entire system is tuned to efficiency, and wasting venom on a target that cannot be consumed represents a significant cost. Within the broader marine ecosystem, the role of sea snakes extends beyond individual predation events. By targeting specific species of fish, they influence population dynamics and contribute to the regulation of prey communities. Their presence can alter the behavior of fish, driving changes in schooling patterns, habitat use, and feeding strategies. These shifts can cascade through the food web, affecting other predators and prey in ways that are not immediately visible. The sea snake, through its precise and efficient use of venom, becomes a subtle but important force in shaping the structure of its environment. The evolutionary pathway that led to this level of specialization involves a series of trade-offs. Transitioning from land to sea required adaptations in respiration, locomotion, and sensory perception. Sea snakes retain lungs, but have extended them along much of their body length, increasing their capacity for oxygen storage and allowing them to remain submerged for extended periods. Some species can even absorb oxygen through their skin, further enhancing their ability to operate in an aquatic environment. These physiological changes support a lifestyle where encounters with prey are intermittent but decisive, aligning with the need for a venom system that can resolve interactions quickly. The relationship between sea snakes and their prey also highlights the ongoing nature of evolutionary arms races. While the venom is highly effective, it is not universally so. Some fish species may exhibit partial resistance, requiring higher doses or more precise delivery to achieve the same effect. This creates selective pressure on both predator and prey, driving incremental changes in toxin composition and physiological defenses. Over time, this dynamic can lead to diversification in venom profiles as different populations adapt to the specific challenges of their local environments. At the molecular level, the toxins produced by sea snakes are the result of gene duplication and modification, processes that allow for the refinement of function over evolutionary time. Small changes in protein structure can significantly alter the binding affinity and specificity of the toxin, enhancing its effectiveness against particular targets. This capacity for variation provides the raw material for natural selection, enabling sea snakes to maintain their edge in a constantly changing ecological landscape. The venom is not static, but a living system, shaped by the interactions it mediates. The apparent simplicity of the sea snake's hunting strategy, approach, strike, withdraw, belies the complexity of the processes involved. Each movement is informed by sensory input, each strike calibrated by experience and physiology, and each outcome determined by a cascade of biochemical interactions. The snake does not need to overpower its prey physically. It only needs to initiate a process that the prey cannot stop. This reliance on internal disruption rather than external force represents a fundamental shift in how predation can be achieved. As the immobilized fish drifts downward, its body unresponsive to the signals that once drove its movement, the sea snake follows at a measured pace. There is no urgency, no need to rush, because the critical moment has already passed. The strike, brief and precise, has set in motion a sequence of events that will inevitably lead to capture. The surrounding water remains in motion, currents shifting, other fish darting through the space. Yet within this dynamic environment, the outcome of this particular interaction is fixed. The silent paralysis strike is not an act of aggression, but an expression of efficiency. It is a system that minimizes effort while maximizing effect, reducing the need for prolonged engagement and conserving valuable resources. In a world where energy and opportunity are tightly constrained, such optimization can mean the difference between survival and failure. The sea snake, through its precise use of venom, demonstrates that dominance in the ocean is not always about force or speed, but about the ability to intervene at the most fundamental level of biological function. where a single molecular interaction can determine the fate of an entire organism. The pressure comes first. The realization comes too late. A grazing fish dips its snout toward what appears to be a harmless patch of spines, or a drifting foot settles briefly against the reef, and in that instant, the surface transforms from scenery into mechanism. Hundreds of rigid needles flex and penetrate, some snapping under force, others remaining embedded, and between them, something smaller begins to close. Microscopic jaws, clamping, injecting, holding. Pain radiates outward, not as a single point, but as a field, a spreading chemical signal that overwhelms local tissue and scrambles nerve responses. Nothing lunged, nothing chased, and yet the interaction carries the same consequence as a strike. The sea urchin does not act. It simply exists, and that existence is weaponized. The sea urchin's body is a paradox of simplicity and sophistication, a spherical architecture built from interlocking calcareous plates that provide structural integrity while supporting a forest of spines that function as both armor and delivery system. Each spine is anchored to the test through a ball and socket joint, allowing it to pivot in response to external stimuli. This mobility is not for locomotion, but for defense, enabling the urchin to orient its spines toward the source of disturbance. The movement is subtle, yet coordinated, a slow realignment that transforms the organism into a reactive barrier, where any point of contact is met with resistance. Beneath this physical defense lies a chemical layer that defines the urchin's true capability. The spines are not merely rigid projections. In many species, they are capable of delivering venom through microscopic channels or through associated structures that release toxins upon breakage. The composition of this venom varies, but it often includes compounds that induce intense pain, disrupt nerve function, and trigger inflammatory responses. In some cases, the toxins can interfere with ion channels in nerve cells, altering the transmission of electrical signals and leading to localized paralysis or prolonged discomfort. The effect is not always immediately lethal, but it is sufficiently disruptive to deter further interaction. Yet the spines are only part of the system. Scattered across the surface of the urchin are pedicellariae, minute, claw-like appendages that operate with a level of precision disproportionate to their size. These structures are capable of detecting contact and responding by snapping shut, gripping onto the offending tissue, and, in certain species, injecting venom directly. The pedicellariae function as independent units, each one a micro-scale defender that can act autonomously. When combined across the entire surface of the urchin, they create a distributed network of sensors and effectors, a living array that converts any touch into a localized chemical response. This dual-layered defense, spines for penetration and pedicellariae for targeted engagement, transforms the urchin into more than a passive organism. It becomes a zone of risk, a region of the reef where interaction carries an inherent cost. Unlike mobile predators that must seek out opportunities, the urchin relies on the inevitability of contact within a crowded ecosystem. Fish graze, currents shift, organisms settle and move, and in this constant flux, the urchin remains fixed, its defenses always active. The absence of movement is not a limitation, but a strategy, one that leverages the density of life around it to ensure that encounters occur without the need for pursuit. The ecological implications of this strategy are profound. Sea urchins are not merely defensive entities. They are active participants in the shaping of their environment. Many species feed on algae, scraping surfaces with specialized mouthparts that can remove significant amounts of vegetation. In doing so, they influence the balance between algal growth and coral dominance, contributing to the structure and composition of reef ecosystems. However, their defensive capabilities also protect them from predation, allowing populations to expand under certain conditions. When predator numbers decline, urchin populations can increase dramatically, leading to overgrazing and the transformation of complex habitats into barren landscapes. This introduces a critical reversal in perception. The sea urchin, often viewed as a static and vulnerable organism, can become an agent of ecological change when its defenses allow it to persist unchecked. The very traits that protect it from individual threats enable it to exert pressure on the system as a whole. In regions where key predators, such as sea otters or certain fish species, are removed, urchin populations can explode, creating what are known as urchin barrens, expanses of reef stripped of algae and reduced in biodiversity. Here, the passive defender becomes a dominant force, not through aggression, but through resilience and persistence. At the physiological level, maintaining this defensive system requires a balance of resources, the production of venom, the growth and repair of spines, and the operation of pedicellariae all incur metabolic costs. Sea urchins meet these demands through their feeding strategies, consuming algae and other organic material to fuel their biological processes. Their ability to thrive in environments with variable resource availability reflects an adaptation to conditions where both food and threats are unpredictable. By investing in a defense that reduces the need for escape or rapid response, they can allocate energy toward growth and reproduction, reinforcing their presence within the ecosystem. The interaction between sea urchins and their predators further illustrates the dynamic nature of this system. Certain species have evolved methods to bypass or tolerate the urchin's defenses. Sea otters, for example, use tools to break open the urchin's test, accessing the softer tissues inside while avoiding the spines. Some fish species have specialized feeding behaviors that allow them to consume urchins without triggering the full defensive response. These adaptations represent a counterbalance, a response to the selective pressure imposed by the urchin's chemical and physical defenses. The hazard zone created by the urchin is not absolute. It is a challenge that drives innovation in those that depend on it as a resource. The spatial distribution of sea urchins within a habitat also reflects their defensive strategy. They often occupy crevices, rocky outcrops, and other structures that provide additional protection, enhancing the effectiveness of their spines and pedicellariae. In these positions, they can extend their defenses into the surrounding space while minimizing exposure to direct attack. The result is a mosaic of microhabitats, each influenced by the presence of urchins and the interactions they mediate. Movement through these areas becomes a calculated process for other organisms, where the cost of contact must be weighed against the benefits of foraging or shelter. At a microscopic level, the toxins produced by sea urchins represent a diverse array of compounds with specific effects on biological systems. Some interfere with cellular signaling pathways, altering the function of nerve and muscle cells. Others trigger inflammatory responses, leading to swelling and pain that can persist long after the initial contact. The diversity of these compounds suggests a history of evolutionary refinement, where different species have developed unique chemical profiles in response to their particular ecological contexts. This chemical diversity contributes to the overall effectiveness of the urchin's defense, ensuring that it can deter a wide range of potential threats. The concept of a permanent hazard zone extends beyond the immediate vicinity of the urchin. The presence of multiple individuals can create larger areas where the risk of contact is elevated, influencing the movement patterns of other species. Fish may avoid certain regions, altering their feeding routes and interactions with other organisms. This, in turn, can affect the distribution of algae, the behavior of predators, and the overall flow of energy within the ecosystem. The urchin's defense, while localized at the level of the individual, scales up to influence broader ecological dynamics. As the initial encounter fades, the consequences linger. Embedded spines may remain in the tissue, continuing to deliver toxins or acting as sites for infection. The affected organism must divert energy toward healing and recovery, resources that could otherwise be used for growth or reproduction. This cost reinforces the deterrent effect of the urchin's defenses, embedding the experience into the behavioral repertoire of the organism. Over time, these individual experiences accumulate, shaping the interactions within the ecosystem and reinforcing the boundaries defined by the presence of the urchin. In the stillness of its form, the sea urchin achieves a level of control that belies its lack of movement. It does not need to pursue or evade. It simply maintains a state of readiness, a constant potential that transforms the surrounding space. The living spike trap is not a momentary threat, but a continuous condition, a presence that defines where and how other organisms can exist. Through the integration of physical structure and chemical defense, the sea urchin turns immobility into a form of power, one that operates quietly yet persistently, shaping the ecosystem, one contact at a time. The current snaps a drifting juvenile into the fringe of soft, waving arms, and the world changes at the scale of a single cell. A hair-thin trigger bends, a capsule explodes, and a filament pierces flesh with a force generated by osmotic pressure that dwarfs the animal's size. In the same instant, dozens more fire, a synchronized volley that turns contact into capture. The fish does not understand the boundary it has crossed, only that movement has become unreliable, that the muscles which propelled it moments ago now answer with hesitation and failure. Around it, the tentacles continue their slow, floral motion, but the calm is deceptive. This is a field already activated, a lattice of sensors and injectors that has converted open water into a trap without edges. A sea anemone is not a flower, though its radial symmetry and petal-like tentacles invite that comparison. It is a predatory surface engineered to translate touch into chemistry. Each tentacle is studded with kinetocytes, specialized cells housing nematocysts that remain cocked under internal pressure until the correct combination of mechanical deflection and chemical cues releases them. This two-key system is critical because the anemone cannot afford indiscriminate discharge. Every nematocyst is a single-use device that must be regenerated at a metabolic cost. The requirement for both touch and the right molecular signature filters out noise, passing debris, sediment, even many non-prey organisms, while ensuring that living, suitable targets trigger a rapid response. What appears to be passivity is, in fact, a tightly regulated decision system operating across thousands of independent units. When the decision resolves, the outcome is immediate. The nematocyst's tubule everts with explosive speed, penetrating the prey's outer layers and delivering a cocktail of toxins directly into the tissues. These toxins vary among species, but often include neuroactive peptides that disrupt ion channel function and interfere with synaptic transmission, along with cytolysins that compromise cell membranes. The dual effect is crucial. Electrical signaling falters as membranes destabilize, converting a mobile organism into a compliant mass that can be manipulated by ciliary currents and muscular contractions. The anemone does not pursue. It converts proximity into inevitability, drawing the immobilized prey toward its central mouth through coordinated movements of its body column and tentacles. The architecture that enables this process is deceptively simple. Anchored to a substrate by a petal disc, the anemone maintains a vertical column topped by an oral disc from which the tentacles radiate. Hydrostatic pressure within the body cavity provides rigidity, allowing the organism to adjust its posture and tentacle spread in response to environmental conditions. In areas of strong current, tentacles may extend further into the flow, increasing the effective capture area and exploiting the movement of water to bring prey into contact. This is not random positioning, but a form of environmental integration where fluid dynamics become an extension of the anemone's reach. The trap field is not fixed. It is tuned to the physics of the surrounding water. Within this field, capture is only the first phase of a longer sequence. Once prey is subdued, the anemone's gastrovascular cavity begins the process of digestion, secreting enzymes that break down tissues externally before absorption. The energy extracted supports not only maintenance and growth, but the continual production of new nematocysts, closing the loop between feeding and defense. The same structures that immobilize prey also deter potential predators, creating a unified system where the distinction between offense and defense dissolves. A grazing fish that brushes too close experiences the same initial barrage as a captured prey item, reinforcing the boundaries of the anemone's domain through pain and dysfunction. Yet the most striking aspect of the anemone's biology is not its capacity for indiscriminate harm, but its ability to discriminate. Among the species that move freely through the tentacles is the clownfish, an organism that appears to violate the fundamental rule of the trap field. It darts, hovers, and feeds within the reach of the stinging cells, without triggering them, a living contradiction that reveals a deeper layer of complexity. The key lies in chemistry, specifically in the mucus that coats the clownfish's body. This mucus either lacks the molecular cues that activate the nematocysts or actively mimics the chemical profile of the anemone itself, effectively masking the fish as part of the host. The cnidocytes, tuned to detect foreign signals, remain inert. This selective response transforms the anemone from a simple predator into a participant in a symbiotic network. The clownfish gains protection from its own predators, which are deterred by the anemone's sting, while the anemone benefits from the presence of the fish. The clownfish's movements increase water circulation around the tentacles, enhancing oxygen exchange and potentially improving the delivery of prey. It may also remove parasites and debris, maintaining the health of the host. In some cases, the fish's waste products provide nutrients that can be absorbed by the anemone or by the symbiotic algae that reside within its tissues. What begins as a chemical exclusion, killing intruders while sparing allies, evolves into a mutually beneficial relationship that integrates multiple biological systems. The presence of symbiotic algae, often dinoflagellates, adds another dimension to this integration. These microscopic partners reside within the anemone's cells, performing photosynthesis and supplying organic compounds that supplement the anemone's diet. In return, The algae receive protection and access to sunlight, positioning the anemone as both predator and producer within the ecosystem. The energy derived from photosynthesis supports the maintenance of the trap field, including the production of nematocysts, linking the anemone's chemical warfare capabilities to the flow of solar energy. This connection underscores a broader principle. Even the most aggressive biological systems are embedded within networks of exchange and cooperation. The ecological influence of the anemone extends beyond its immediate interactions. By capturing small fish and invertebrates, it contributes to the regulation of local populations, while its symbiotic relationships create microhabitats that support additional species. The presence of anemones can alter the distribution of organisms within a reef, as some species are drawn to the protection they offer, while others avoid the risk they pose. This creates a mosaic of zones defined by varying levels of danger and refuge, shaping the spatial structure of the ecosystem. The trap field is not an isolated phenomenon, but a node within a larger web of interactions. A reversal emerges when considering the anemone's own vulnerabilities. Despite its potent defenses, it is not immune to predation. Certain species, such as specialized fish and invertebrates, have evolved resistance to the toxins or behaviors that allow them to feed on the anemone without triggering its full defensive response. In these cases, the predator becomes the one organism that can navigate the trap field, turning the anemone's strength into a point of exploitation. This dynamic illustrates the limits of even highly specialized systems and highlights the ongoing nature of evolutionary adaptation, where each advantage invites a countermeasure. At the molecular level, the specificity of the anemone's toxins and triggers reflects a history of coevolution with its environment. Variations in toxin composition among species suggest adaptation to different prey types and ecological conditions, while differences in nematocyst sensitivity indicate fine-tuning of the detection mechanisms. These variations contribute to the diversity of anemones and their roles within different habitats, from shallow reefs to deeper, less illuminated environments. The trap field is not a single design, but a family of solutions, each calibrated to the demands of its context. The apparent stillness of the anemone masks a continuous process of sensing, responding, and renewing. Tentacles extend and retract, nematocysts are discharged and replaced, and chemical gradients shift with the flow of water. The organism remains anchored, yet it is dynamically engaged with its surroundings, converting the random movement of the environment into structured interactions. This capacity to harness external motion reduces the need for internal expenditure, aligning with the broader theme of efficiency that characterizes many sessile organisms. The anemone does not need to move to influence its world. It shapes that world by defining the terms of contact. As the immobilized fish is drawn inward, its brief encounter with the trap field concludes, but the implications persist. Other organisms, sensing the disturbance or having learned from prior encounters, adjust their behavior, altering their paths and interactions. The boundaries of the anemone's domain are reinforced not by visible markers, but by the accumulation of experiences encoded in the nervous systems of those that survive. Over time, these behavioral patterns contribute to the stability of the ecosystem as species partition space and resources in response to the risks and opportunities presented by organisms like the anemone. In this way, the sea anemone exemplifies a form of selective chemical warfare that transcends simple predation. It does not merely capture and consume, it discriminates, integrates, and influences. Its tentacles define a space where the rules of engagement are dictated by molecular recognition. where allies are permitted and intruders are neutralized. The living trap field is both a boundary and a bridge, a site where conflict and cooperation coexist, and where the quiet application of chemistry shapes the flow of life around it. The tentacles should kill it. They close, brush, recoil. Thousands of microscopic harpoons primed to fire on contact, yet the small orange body darts through them untouched, weaving in and out of a lethal forest as if the rules no longer apply. A predator approaches, cautious but hungry, tracking the clownfish's movement, and then it stops, not because of the fish, but because of the space the fish occupies. The anemone's reach is absolute, its chemical field unforgiving, and yet within that boundary, the clownfish moves freely, untouchable not by strength, not by speed, but by something far more precise. The danger is still there. The difference is that one organism has rewritten how that danger recognizes it. The sea anemone's defense system is built on detection, not indiscriminate attack. Each nematocyst, those microscopic, coiled injectors, requires a specific combination of mechanical disturbance and chemical identity to trigger. This is a necessary constraint because firing blindly would waste valuable resources, leaving the organism vulnerable until those cells are replaced. The system is selective by design, tuned to recognize prey and threats through molecular cues present on the surface of other organisms. Most fish that brush against the tentacles activate this system instantly, triggering a cascade of toxin delivery that paralyzes or deters them. The clownfish does not. The key lies in its mucus, a thin, dynamic layer that coats its skin and serves as both shield and disguise. Unlike the mucus of other fish, which often contains compounds that activate nematocysts, the clownfish's coating lacks these triggers or actively mimics the chemical signature of the anemone itself. This is not passive resistance, it is biochemical deception. By presenting itself as chemically familiar, the clownfish bypasses the anemone's detection system entirely. The nematocysts remain inert, not because they are incapable of firing, but because they do not perceive a target. The weapon is still loaded. It simply does not recognize the clownfish as something worth attacking. This relationship is not instantaneous. Juvenile clownfish must undergo a process of acclimation, a gradual introduction to the anemone's tentacles, during which they make brief, tentative contacts. Each interaction allows the fish to incorporate elements of the anemone's mucus or adjust its own chemical profile, refining the disguise until it is fully accepted. This behavior reflects a sophisticated interaction between physiology and learning, where the fish actively participates in shaping its own immunity. The process is risky. A misstep can result in envenomation, but once established, the protection is profound, granting the clownfish access to a space that is effectively off-limits to most other organisms. Within this space, the clownfish gains more than just safety. The anemone becomes a fortress, its tentacles forming a barrier that deters predators unable to penetrate the chemical defense. Larger fish that might easily consume the clownfish are forced to remain at a distance, constrained not by the prey itself, but by the environment it inhabits. This creates a reversal in the traditional predator-prey dynamic. The clownfish, small and otherwise vulnerable, occupies a position of relative security, while the predator, larger and more powerful, is excluded. The hierarchy is inverted not through direct confrontation, but through the manipulation of another organism's defensive system. The benefits to the clownfish extend beyond protection. Living within the anemone provides access to food resources, including small invertebrates and organic particles that drift into the tentacles. The fish also feeds on parasites and debris that accumulate on the anemone, contributing to the host's health. This interaction is not one-sided. It is a mutualistic relationship where both participants derive advantage. The clownfish's movements increase water circulation around the anemone, enhancing gas exchange and potentially improving the delivery of nutrients. In some cases, the fish's waste products provide nitrogenous compounds that support the growth of symbiotic algae within the anemone's tissues, linking the relationship to broader cycles of energy and nutrient flow. This integration of systems highlights a deeper principle. Survival in complex ecosystems often depends on the ability to navigate and exploit existing networks, rather than relying solely on individual capabilities. The clownfish does not evolve its own venom or develop physical defenses comparable to those of larger species. Instead, it leverages the chemical infrastructure of the anemone, effectively outsourcing its protection. This strategy reduces the need for costly adaptations, allowing the fish to allocate energy toward growth, reproduction, and other functions. It is a form of efficiency achieved through association, where the boundaries between organisms become blurred. The ecological implications of this relationship are significant. The presence of clownfish can influence the distribution and behavior of other species within the reef. Predators may avoid areas dominated by anemones, altering their hunting patterns and potentially affecting the populations of other prey species. At the same time, the clownfish's activities can impact the health and growth of the anemone, which in turn affects the organisms that depend on it. This creates a network of interactions that extends beyond the immediate partnership, shaping the structure and dynamics of the ecosystem. Yet this system is not without its limitations. The clownfish's reliance on the anemone ties its fate to that of its host. Changes in environmental conditions that affect the anemone, such as temperature fluctuations, pollution, or habitat destruction, can have direct consequences for the fish. The very specialization that provides protection also imposes constraints, restricting the clownfish to specific habitats and making it vulnerable to disruptions in those environments. This introduces a subtle vulnerability within the apparent strength of the strategy, a reminder that specialization often comes with trade-offs. From an evolutionary perspective, the relationship between clownfish and anemones represents a co-evolutionary process where both species have adapted in response to each other. The anemone's selective detection system creates an opportunity for organisms that can bypass it, while the clownfish's presence provides benefits that reinforce the partnership. Over time, these interactions can lead to increased specialization, with different species of clownfish associating with specific types of anemones. This diversification reflects the complexity of the underlying mechanisms, where slight variations in chemical signaling can determine compatibility and success. At the molecular level, the clownfish's mucus is not static but dynamically regulated, adjusting its composition in response to environmental cues and interactions with the anemone. This flexibility allows the fish to maintain its disguise, even as conditions change, ensuring continued protection. The ability to modulate its chemical profile represents a form of physiological plasticity, enabling the clownfish to adapt to different hosts or conditions within certain limits. This adaptability is a key component of its success, allowing it to exploit a niche that would otherwise be inaccessible. The concept of immunity as a weapon challenges traditional notions of defense and offense. In the case of the clownfish, survival is not achieved through the ability to inflict damage, but through the capacity to avoid it entirely. By integrating itself into the anemone's chemical system, the fish effectively removes itself from the category of potential targets. This is not invisibility in the visual sense, but in the chemical domain, where recognition determines response. The clownfish does not need to outrun predators or outfight them. It simply occupies a space where they cannot follow. As the predator withdraws, deterred by the unseen boundary of the anemone's reach, the clownfish continues its movement, unaffected by the tension that has just unfolded. The tentacles sway, the nematocysts remain primed, and the chemical field persists, unchanged. Within this environment, the clownfish exists not as an anomaly, but as a participant in a system that it has learned to navigate and manipulate. The danger has not diminished. It has been redirected, shaping the interactions of all who approach. In this way, the clownfish exemplifies a form of survival that is both subtle and profound. It demonstrates that the most effective strategies are not always those that confront threats directly, but those that redefine the conditions under which those threats operate. By weaponizing immunity, the clownfish transforms a lethal environment into a sanctuary, leveraging the defenses of another organism to secure its own place within the ecosystem. The result is a delicate balance, a partnership sustained by chemistry and behavior, where the line between ally and weapon becomes indistinguishable. The transformation happens before the threat can decide what it is seeing. A shape on the seafloor fractures into motion, limbs unfurling, flattening, elongating, until the single animal dissolves into something else entirely. A striped ribbon gliding with the cadence of a venomous serpent, or a fan of spines radiating outward in a warning display that should not belong here. The predator pauses, its approach interrupted not by force, but by uncertainty. The strike that seemed inevitable dissolves into hesitation, and hesitation in the open ocean is often the difference between action and retreat. Nothing has been injected, nothing has been fired, and yet the encounter has already shifted. The mimic octopus has not attacked, it has rewritten perception. The mimic octopus operates at the intersection of biology and information, exploiting the fact that survival is often determined not by what is true, but by what is believed. Its body is a dynamic canvas, composed of muscle fibers, capable of altering texture, shape, and color with remarkable precision. Chromatophores expand and contract to produce rapid shifts in pigmentation, while deeper layers of iridophores and leucophores manipulate light reflection, creating patterns that can mimic the appearance of entirely different species. This is not simple camouflage, where the goal is to disappear into the background. It is active impersonation, a deliberate projection of signals that correspond to organisms known to be dangerous. The success of this strategy depends on the reliability of those signals within the ecosystem. Venomous species, such as sea snakes and lionfish, have established reputations through repeated interactions with predators, where the cost of attack is high and often immediate. Over time, predators learn to associate specific visual patterns, movements, and even spatial behaviors with danger. These associations are encoded within their neural circuits, shaping decision-making processes that favor avoidance over risk. The mimic octopus taps into this existing framework, presenting itself as a member of these dangerous categories without possessing the underlying chemical defenses. This is where the concept of chemical reputation becomes critical. The mimic octopus does not produce venom, but it leverages the ecological memory of venomous species. Its displays are effective because they align with the expectations of the observer, triggering avoidance behaviors that have been reinforced through prior experience or inherited predispositions. The octopus is, in effect, borrowing the chemical history of other organisms, translating it into a visual and behavioral language that predators understand. This is deception not at the level of molecules, but at the level of cognition, where perception dictates response. The mechanics of this deception are highly context-dependent. The mimic octopus does not adopt a single form, but selects from a repertoire of possible identities based on the nature of the threat. In the presence of a predator that hunts along the seafloor, it may flatten its body and extend its arms in a pattern that resembles a venomous flatfish or sea snake, moving in a way that reinforces the illusion. When confronted by a different type of predator, it may shift to a configuration that mimics the spines and coloration of a lionfish, presenting a profile that suggests toxicity and physical deterrence. This flexibility allows the octopus to tailor its response, increasing the likelihood that the deception will be effective. Underlying this adaptability is a sophisticated nervous system capable of rapid processing and decision-making. The octopus must assess the type of threat, select an appropriate mimicry pattern, and execute the transformation within a time frame that prevents the predator from acting. This requires integration of sensory input, memory, and motor control at a level that rivals more traditionally recognized intelligent organisms. The mimicry is not a fixed trait, but a behavior that can be adjusted in real time, reflecting a level of control that transforms the octopus from a passive participant into an active manipulator of its environment. The ecological implications of this strategy extend beyond individual encounters. By reducing the likelihood of predation, the mimic octopus can occupy habitats that might otherwise be too risky, expanding its ecological niche. Its presence influences the behavior of predators, which must navigate an environment where appearances can no longer be trusted. This introduces a layer of complexity into predator-prey interactions, where decision-making becomes a balance between caution and opportunity. The cost of a mistake, attacking a truly venomous organism, remains high, reinforcing the effectiveness of the mimic's deception. Yet this system is not without its vulnerabilities. The success of mimicry depends on the persistence of the models it imitates. If the populations of venomous species decline or their signals become less reliable, the effectiveness of the mimic's strategy may diminish. Additionally, predators that have not encountered the model species or that are capable of distinguishing subtle differences may not respond to the deception. This creates a dynamic environment where the mimic octopus must continuously adapt, refining its displays to match the expectations of its audience. This introduces a narrative reversal that challenges the initial perception of the mimic octopus as a purely deceptive entity. While it appears to dominate through illusion, it is in fact dependent on the presence and persistence of other species. Its power is borrowed, not intrinsic, and its survival is tied to the ecological context that gives meaning to its signals. The absence of venom is not simply a lack, it is a strategic choice that relies on the existence of venomous organisms to provide the necessary framework for deception. In this sense, the mimic octopus is part of a larger network, where its identity is shaped by the interactions of multiple species. At a deeper level, the mimic octopus exemplifies a form of evolutionary innovation that prioritizes efficiency. Producing venom is metabolically expensive, requiring the synthesis of complex compounds and the maintenance of specialized delivery systems. By avoiding this cost and instead investing in neural and morphological flexibility, the octopus achieves a similar level of protection through different means. This trade-off reflects a broader principle in evolutionary biology, where multiple pathways can lead to comparable outcomes, each shaped by the constraints and opportunities of the environment. The relationship between the mimic octopus and its predators also highlights the role of learning and memory in shaping ecological interactions. Predators that have experienced the consequences of attacking venomous species carry that information forward, influencing their responses to future encounters. The mimic octopus exploits this memory, presenting cues that trigger avoidance, even in the absence of actual danger. Over time, this can lead to a feedback loop where the presence of mimics reinforces the avoidance behavior, further reducing the likelihood of predation for both the mimic and the model species. The sensory environment in which this interaction occurs is complex, involving not only visual cues, but also chemical and mechanical signals. While the mimicry is primarily visual, it may be supported by behaviors that align with the expected patterns of the model species, such as specific movements or postures. This multi-layered approach increases the robustness of the deception, ensuring that it remains effective across different contexts and conditions. The octopus does not rely on a single signal, but integrates multiple elements to create a convincing illusion. As the predator withdraws, the mimic octopus relaxes its form, its body flowing back into a more neutral configuration that blends with the substrate. The transformation is complete, and the immediate threat has passed, but the implications remain. The predator has been influenced not by force, but by information, its behavior altered by the perception of risk rather than the experience of it. This subtle manipulation underscores the power of deception as a survival strategy, where the ability to control what others believe can be as effective as any physical weapon. In the broader context of the ecosystem, the mimic octopus represents a shift in how we understand defense and predation. It demonstrates that the boundaries between these roles are not fixed, but can be navigated through the manipulation of signals and perceptions. The octopus does not need to overpower its predators or outpace them. It only needs to convince them that it is something they cannot afford to challenge. This is chemical warfare without chemicals, a strategy built on the reputations of others and the cognitive frameworks of those that observe it. The deception chemist does not synthesize toxins or deliver stings, yet it achieves a level of protection that rivals the most chemically armed organisms in the ocean. Its laboratory is the ecosystem itself, its reagents, the interactions and memories of other species, and its product a shifting identity that adapts to the demands of the moment. In a world where survival often depends on the ability to act quickly and decisively, the mimic octopus introduces a different paradigm, one where the most effective action is sometimes the illusion of something else entirely. The shell closes a fraction too late. A crab's claws snap inward on empty water as a soft, shifting shadow resolves into 8 arms that do not rush, do not thrash, but place themselves with unsettling accuracy around the prey's blind angles. There is no visible struggle, only a brief, deliberate contact at the hinge. A puncture so small it is almost theoretical, and then the resistance drains from the crab's body, as if a switch has been thrown somewhere deep within its circuitry. Muscles that should clamp harder instead slacken, reflexes falter, and the animal that moments ago embodied armor and force becomes compliant, unresisting. The octopus does not overpower. It edits the outcome, inserting chemistry at the exact point where control resides. Octopus venom, in its simplest description, is a paralytic cocktail, a mixture of neuroactive compounds capable of interfering with ion channels and synaptic transmission. But in practice, it is something more nuanced, a modular tool deployed by an organism whose nervous system is distributed, plastic, and capable of rapid decision-making. Many species produce saliva containing toxins that can immobilize crustaceans and fish, and in some, such as the blue-ringed lineage, symbiotic bacteria provide tetrodotoxin, amplifying potency to levels that can silence vertebrate nervous systems. Yet across the diversity of octopuses, the unifying principle is not the chemical itself, but the precision with which it is applied. The venom is not sprayed, not broadly delivered. It is injected with intent into locations that maximize effect while minimizing cost. This precision begins with approach. An octopus does not rely on speed in the way pelagic predators do. It relies on ambiguity. Chromatophores expand and contract to break up outlines. Papillae alter skin texture to mimic rock, algae, or coral, and posture shifts to match the geometry of the environment. Each adjustment is both concealment and communication, an alignment with the sensory expectations of other organisms that reduces detection probability. The animal moves not as a single entity, but as a sequence of localized decisions, arms testing, probing, pausing, while the central brain and peripheral ganglia integrate information about texture, chemistry, and movement. The approach is a negotiation with perception, where the goal is not to remain unseen in an absolute sense, but to remain unrecognized as a threat. When contact is made, it is rarely accidental. Suckers along the arms are not merely adhesive structures. They are chemosensory organs, capable of tasting the environment, sampling molecular signatures that inform the octopus about the identity and condition of its prey. This distributed sensing allows the animal to map the target in real time, identifying joints, softer tissues, and points of vulnerability. In crustaceans, the exoskeleton presents a formidable barrier, but it is not uniform. At the articulations, where segments meet, the cuticle thins to allow movement, creating entry points that the octopus can exploit. The beak, a hard, chitinous structure at the center of the arm crown, is then positioned with remarkable accuracy, delivering a bite that penetrates just enough to introduce venom into the internal environment. The chemistry that follows is targeted disruption. Neurotoxins interfere with voltage-gated channels and synaptic receptors, preventing the propagation of action potentials that coordinate muscle contraction, while other components may facilitate the breakdown of connective tissues, easing the process of consumption. The effect is not explosive, it is controlled. The prey's defensive behaviors diminish, its capacity to resist fades, and the octopus maintains a firm but economical grip, avoiding unnecessary exertion. In this way, the venom functions as a force multiplier, allowing a relatively soft-bodied organism to neutralize prey that would otherwise be dangerous or inaccessible. The strategic dimension emerges in the variability of this process. Octopuses can adjust their behavior based on prey type, size, and context, sometimes opting for rapid envenomation, other times employing a slower approach that minimizes risk. This flexibility reflects an underlying cognitive architecture that supports problem-solving and learning. Individuals can remember successful techniques, refine their methods, and even innovate, as seen in behaviors where octopuses drill through shells or manipulate objects to access food. The venom is integrated into this behavioral repertoire, not as a fixed response, but as one option among many, selected when it offers the most efficient path to success. This introduces a reversal that challenges the initial impression of venom as the primary weapon. The toxin, potent as it is, becomes secondary to the intelligence that deploys it. Without the ability to choose the right moment, the correct angle, and the optimal target, the chemical would be far less effective. The octopus's apparent vulnerability, its lack of a rigid skeleton or protective armor, drives the evolution of a system where information processing compensates for physical limitations. Softness becomes an advantage, allowing the animal to compress its body, infiltrate narrow spaces, and position itself in ways that rigid predators cannot, all while maintaining control over the interaction. The environment in which this occurs is not passive. Reefs and rocky substrates are complex, with crevices, overhangs, and variable lighting that create a mosaic of opportunities and constraints. The octopus navigates this complexity by integrating visual input with tactile and chemical cues, constructing a functional map that guides its movements. This mapping is dynamic, updated with each interaction, and informs not only hunting, but also avoidance of predators. Venom, in this context, is not only a tool for feeding, but a component of a broader strategy that includes escape, concealment, and the manipulation of space. At the level of the ecosystem, the octopus's behavior has implications that extend beyond individual predation events. By targeting specific prey, it influences population dynamics, potentially reducing the abundance of certain species, while allowing others to flourish. This selective pressure can contribute to trophic cascades, where changes at one level of the food web propagate through others, altering the composition and function of the community. The precision of the octopus's hunting, guided by its intelligence, means that these effects are not random but patterned, reflecting the animal's preferences and adaptations. There is also an energetic dimension to consider. Venom production is costly, requiring the synthesis of complex molecules and the maintenance of specialized glands. By using venom strategically, the octopus minimizes waste, deploying it only when the expected return, in terms of captured prey, justifies the expenditure. This efficiency is further enhanced by the animal's ability to handle prey carefully, avoiding damage to its own tissues and reducing the risk of injury. The combination of chemical and behavioral economy allows the octopus to operate effectively in environments where resources may be unevenly distributed. The relationship between octopuses and their prey is not static. Some prey species may develop behaviors or physiological traits that reduce vulnerability, such as thicker exoskeletons or more effective escape responses. In turn, octopuses may refine their techniques, targeting new entry points or adjusting their approach. This ongoing interaction represents an evolutionary dialogue where each side responds to the pressures imposed by the other. The venom itself may diversify, with variations in composition, reflecting the need to overcome different types of resistance. The system is dynamic, shaped by continuous feedback between predator and prey. In moments where the octopus becomes the hunted, the same principles apply. Venom can serve a defensive role, deterring predators through the risk of envenomation, But more often, the animal relies on its ability to disappear, to alter its appearance and behavior in ways that reduce the likelihood of detection. Here again, intelligence mediates the use of chemistry, determining when to engage and when to avoid. The decision-making process is fluid, responsive to immediate conditions, and informed by prior experience. As the crab's body yields, its movements stilled not by force, but by interruption of its own internal signaling, the octopus begins the process of consumption, its arms coordinating with a level of autonomy that reflects the distributed nature of its nervous system. Each arm contributes to positioning and manipulation, while the central brain oversees the overall sequence, adjusting as needed. The event, which began with a brief, almost imperceptible contact, unfolds into a complete transfer of energy, one organism converted into another through a chain of decisions and reactions that span scales from the molecular to the behavioral. The combination of venom and intelligence in the octopus represents a synthesis of chemical and cognitive strategies, where neither alone would suffice. The toxin provides the means to neutralize, but it is the timing, placement, and context that determine success. In a world where encounters are fleeting and opportunities must be seized without error, this integration offers a powerful advantage. The octopus does not simply possess a weapon, it understands, in a way that is both instinctive and learned, how to use it with precision. In this sense, the octopus operates less like a brute force predator and more like a system that edits interactions at critical points, introducing small changes that cascade into decisive outcomes. The venom is the catalyst, the intelligence the guide, and together they form a method of predation that is as subtle as it is effective. The result is a form of control that does not rely on dominance in the traditional sense, but on the ability to intervene at the exact moment when the balance can be tipped, transforming potential into inevitability with minimal expenditure. The strike misses by less than the width of an eye. A shadow accelerates from below, jaws opening in a trajectory that has already predicted the prey's escape vector, and then the water fractures, not with force, but with information collapsing into noise. A cloud erupts, dense and dark, but more importantly, alive, with molecules that begin rewriting the sensory field itself. Vision fails first, edges dissolving into uniform darkness, and then the chemical gradients that guide pursuit begin to flatten, distort, and mislead. The predator surges forward into absence, its momentum intact, but its certainty gone. And in that fraction of disorientation, the squid is no longer where it should be. The attack continues, but the target has been erased from the predator's perception before it could be secured in reality. Squid do not defend themselves through brute resistance or chemical lethality. They defend themselves by destabilizing the very systems predators rely on to hunt. Their ink is often mistaken as a simple visual screen, a smokescreen equivalent that obscures line of sight. But this interpretation captures only a portion of its function. At the level of chemistry, squid ink is a complex suspension of melanin granules, mucus, and bioactive compounds that together create a multi-layered disruption. The melanin provides opacity, absorbing and scattering light to eliminate contrast, while the mucus increases the persistence and cohesion of the cloud, allowing it to maintain structure in a medium that would otherwise disperse it rapidly. This combination ensures that the visual field is not merely dim, but fundamentally altered, removing the cues necessary for accurate targeting. Yet the deeper layer of this defense lies in the chemical signals embedded within the ink. Some species produce compounds that interact with the olfactory systems of predators, effectively saturating or confusing the receptors that detect prey-derived cues. In aquatic environments, where chemical gradients are often as important as visual ones, this interference can be decisive. Predators track prey through subtle differences in dissolved molecules, following trails that lead them through complex spatial environments. When these gradients are disrupted, the path collapses, and the predator is left navigating without reliable guidance. The squid does not need to outrun its pursuer indefinitely. It only needs to break the chain of information that connects the predator to its target. The release of ink is tightly coupled with the squid's locomotion. As the cloud expands, the animal executes a rapid, jet-propelled escape, expelling water through its siphon to generate thrust. This movement is not random, but directed, often taking advantage of the momentary blindness imposed on the predator. The timing is critical. Ink released too early dissipates before it can be effective. Too late, and the predator may already have established a firm trajectory. The squid's nervous system integrates sensory input with motor output to coordinate this sequence, ensuring that the chemical and physical components of the escape operate in synchrony. The result is not a simple retreat, but a controlled disengagement, where the squid manipulates the predator's perception to create a window of opportunity. In some cases, the ink cloud does more than obscure. It mimics. By shaping the release and combining it with mucus, the squid can form a pseudomorph, a denser mass that approximates the size and movement of the squid itself. This decoy drifts or moves in a way that draws the predator's attention, effectively replacing the real target with a temporary illusion. The predator, already operating under compromised sensory conditions, may commit to this false target, expending energy and time while the squid escapes in a different direction. This introduces a layer of deception that complements the chemical disruption, turning the ink from a passive barrier into an active component of misdirection. The compounds within the ink also have subtler effects on predator behavior. Research suggests that certain molecules can act as phagomimetic agents, triggering feeding responses or interfering with normal predatory patterns. In essence, the ink may not only hide the squid, but also manipulate the predator's internal state, shifting it from a focused pursuit to a more generalized or confused response. This biochemical interaction represents a form of sensory warfare that extends beyond the external environment into the neural processing of the predator itself. The battlefield is not just the water around the squid, it is the predator's perception and decision-making apparatus. From an energetic perspective, this strategy reflects a balance between cost and benefit. Producing ink requires resources, and the reserves are finite, stored within an ink sac that must be replenished over time. The squid cannot afford to deploy this defense indiscriminately. Instead, it must assess the level of threat and determine whether the expenditure is justified. This decision-making process is influenced by factors such as predator size, distance, and the availability of alternative escape routes. The selective use of ink ensures that it remains an effective tool, reserved for situations where the probability of capture is high and other strategies are insufficient. The ecological implications of this defense extend into the behavior of both squid and their predators. Predators that frequently encounter ink-releasing prey may develop strategies to compensate, relying more heavily on other senses or adjusting their attack patterns to minimize the effectiveness of the ink. This can lead to an ongoing cycle of adaptation, where improvements in squid defense are met with refinements in predator tactics. At the same time, the presence of ink clouds in the environment may influence the behavior of other organisms, creating temporary zones of reduced visibility and altered chemical composition that affect multiple species simultaneously. A reversal emerges when considering the apparent vulnerability of the squid. Soft-bodied and lacking the protective structures of many marine organisms, it would seem to be at a disadvantage in a world of sharp teeth and powerful jaws. Yet this very lack of rigidity enables the flexibility required for its escape strategy. The squid's body can change shape rapidly, compressing and expanding as needed, while its nervous system supports the rapid integration of sensory and motor functions. The absence of armor is not a weakness, but a condition that allows for the development of a different kind of defense, one that operates through manipulation rather than resistance. The integration of visual and chemical disruption also highlights the complexity of the squid's interaction with its environment. Light and dissolved molecules behave differently in water, each subject to distinct physical constraints. Yet the squid's ink exploits both simultaneously. By altering the optical properties of the water, it removes the spatial cues necessary for visual tracking. By modifying the chemical composition, it disrupts the gradients that guide olfactory navigation. This dual interference creates a compounded effect, where the loss of one sense is reinforced by the impairment of another, amplifying the overall impact on the predator's ability to respond. At the level of the ecosystem, squid occupy a position that links multiple trophic levels, serving as both predator and prey. Their ability to evade capture influences the distribution of energy within the system, affecting the success rates of higher-level predators and the survival of the squid themselves. This dynamic contributes to the balance of marine communities, where the efficiency of predation and the effectiveness of escape strategies are constantly negotiated. The squid's ink, as a tool of escape, plays a role in this balance, shaping the outcomes of countless interactions across time. As the cloud begins to dissipate, its structure breaking down under the influence of currents and diffusion, the immediate chaos resolves into a new equilibrium. The predator, having lost its target, must recalibrate, its internal models of the environment updated to reflect the disruption. The squid, now at a distance, resumes its movement through the water, its body once again blending into the shifting patterns of light and shadow. The encounter leaves no lasting mark on the physical environment, yet it alters the behavior of the participants, reinforcing the strategies that led to survival. In this system, the squid does not confront its predators directly. It undermines them. By targeting the sensory foundations of predation, it transforms the interaction from a contest of speed or strength into one of information control. The chemical escape system is not about overpowering an opponent, but about rendering the opponent's tools ineffective, turning certainty into doubt and precision into approximation. The battlefield becomes chaotic, not through random disturbance, but through deliberate manipulation of the signals that define it. The strike is already finished. Before the shock wave arrives, a blur of color compresses, releases, and the water itself fractures. Pressure spikes. A cavitation bubble forms, collapses, and flashes with a brief, searing pulse of light that exists for less than the time it takes. A nerve impulse to travel a fingertip. The shell that moments ago resisted every attempt at entry now splits along lines it did not know it had, not because it was crushed, but because the physics of water were forced to betray it. The mantis shrimp does not simply hit, it weaponizes the medium around it, turning liquid into a transient hammer that multiplies the force of its own motion. This physical phenomenon is only the most visible layer of a system that operates across multiple domains of perception. The mantis shrimp's raptorial appendages, specialized limbs that store elastic energy and release it with extraordinary translated into acceleration that can exceed the tolerances of biological tissue, are often framed as the defining feature of the animal. Yet the strike itself is a culmination of a sensory and computational process that begins long before contact. The shrimp's compound eyes, among the most complex in the animal kingdom, are divided into multiple regions, each capable of detecting different wavelengths and polarizations of light. This configuration allows the mantis shrimp to perceive aspects of the environment that are invisible to most other organisms, including patterns of polarized light and subtle variations in color that carry information about the identity and state of other animals. These visual capabilities are not isolated from chemical sensing. They are integrated into a broader system that interprets both optical and molecular cues. Fluorescence, the emission of light at longer wavelengths after absorption of higher energy photons, plays a role in communication and possibly in intimidation. Certain species possess fluorescent markings that become visible under specific lighting conditions, creating signals that can be detected by conspecifics or rivals with similarly tuned vision. These signals may convey information about species identity, reproductive status, or territorial boundaries, functioning as a form of communication that operates outside the perceptual range of many predators and prey. At the same time, chemical cues, pheromones, and other signaling molecules circulate through the water, providing another layer of information. The mantis shrimp's ability to detect and interpret these cues allows it to construct a multidimensional representation of its surroundings, where visual and chemical data are combined to inform behavior. This integration is critical for an organism that must navigate a complex environment filled with both opportunities and threats. The decision to strike, to display, or to retreat is not based on a single input, but on a synthesis of signals that together define the context of the interaction. The concept of hybrid warfare emerges from this integration. The mantis shrimp does not rely solely on its physical strike to dominate. It uses its sensory capabilities to shape the conditions under which that strike occurs. By detecting signals that others cannot, it gains an informational advantage, allowing it to anticipate movements, identify vulnerabilities, and choose moments of engagement with precision. This is not merely a matter of reaction time, but of predictive modeling, where the shrimp's nervous system processes incoming data to generate expectations about the behavior of other organisms. The strike, when it comes, is the execution of a decision that has already been refined through layers of sensory input. This system extends into interactions with rivals. Mantis shrimp are known for their territorial behavior, defending burrows that serve as both shelter and hunting platforms. Encounters between individuals can escalate into displays that involve not only physical posturing, but also the exhibition of visual signals that communicate strength and intent. Fluorescent patterns may be revealed, appendages raised, and movements exaggerated, creating a display that is as much about perception as it is about potential force. In many cases, these displays resolve the conflict without physical contact, as one individual assesses the signals and withdraws, avoiding the cost of injury. Here, the narrative reverses. The mantis shrimp, often portrayed as an unstoppable force due to its striking ability, reveals a reliance on communication and perception that reduces the need for actual combat. The devastating punch becomes a last resort, a tool that is used when other strategies fail. The true dominance lies in the ability to control the interaction before it reaches that point, to influence the decisions of others through signals that convey information about risk and reward. The physical weapon is supported and often superseded by a system that operates at the level of cognition and interpretation. The ecological context in which this occurs is equally important. Coral reefs and similar habitats are dense with life, creating an environment where encounters are frequent and the cost of miscalculation is high. The mantis shrimp's ability to process complex sensory information allows it to navigate this environment with a degree of precision that reduces unnecessary conflict and maximizes successful predation. By selecting appropriate targets and timing its strikes effectively, it conserves energy and minimizes exposure to its own predators. The cavitation effect produced by the strike introduces an additional layer of complexity. The rapid acceleration of the appendage creates a low-pressure zone in the water, leading to the formation of a vapor bubble that collapses with sufficient to generate a secondary shockwave and a brief flash of light. This phenomenon not only amplifies the physical impact, but also contributes to the sensory experience of the interaction. The sudden burst of light and pressure may disorient prey, adding a momentary disruption that complements the mechanical damage. In this way, the mantis shrimp's strike operates across physical and sensory domains, combining force with environmental manipulation. From a physiological perspective, maintaining such a system requires significant investment. The structures that store and release elastic energy must withstand repeated, and the sensory apparatus must process large volumes of data in real time. The neural architecture that supports this integration is adapted to handle both rapid and more complex decision-making processes, reflecting a balance between and flexibility. This balance is critical, as the shrimp must respond quickly to immediate threats, while also adjusting its behavior based on changing conditions and prior experiences. The interaction between mantis shrimp and their prey also illustrates the ongoing nature of evolutionary adaptation. Prey species may develop thicker shells, faster escape responses, or behaviors that reduce exposure to strikes. In turn, mantis shrimp may refine their targeting, adjust their timing, or alter their strategies to maintain effectiveness. This dynamic creates a feedback loop where each side influences the driving incremental changes that shape the characteristic of both predator and prey. At the level of the ecosystem, the mantis shrimp's activities contribute to the redistribution of energy and the structuring of communities. By preying on certain species, it influences population dynamics, which can cascade through the food web. Its territorial behavior also affects the spatial distribution of organisms, as areas around its burrow become zones of influence where other species adjust their movements and interactions. The combination of physical and sensory control allows the mantis shrimp to exert an impact that extends beyond individual encounters. The concept of warfare, in this context, expands beyond the idea of direct confrontation. It encompasses the manipulation of information, the control of perception, and the strategic deployment of within a framework defined by sensory input and environmental conditions. The mantis shrimp exemplifies this broader definition, operating not just as a striker, but as an interpreter and influencer of the signals that govern behavior in the marine environment. As the settles after the strike, the immediate violence gives way to a quieter form of control. The mantis shrimp retreats to its burrow, its eyes continuing to scan the surroundings, processing light and motion in ways that few other organisms can match. The environment remains dynamic, filled with interactions, and yet within this complexity, the mantis shrimp maintains a position defined not only by its ability to deliver force, but by its capacity to shape the conditions under which that force is applied. In the end, the mantis shrimp's hybrid warfare is not about overwhelming the opponent through sheer power or chemical toxicity alone. It is about integrating multiple systems, mechanical, optical, and chemical, into a cohesive strategy that maximizes effectiveness while minimizing. By controlling perception, it reduces the need for constant, allowing it to operate with a level of efficiency that reflects the sophistication of its adaptations. The battlefield is not just the space where organisms collide, it is the network of signals and interpretations that determine how and when those collisions occur. The sting should end the encounter, not begin a transformation. A translucent ribbon glides across the reef and deliberately presses its soft tissue into the reach of a tentacle that would immobilize almost anything else. And in that instant, a barrage of microscopic harpoons discharges into living skin. Yet the animal does not recoil, does not convulse, does not surrender control. Instead, the contact becomes acquisition. Capsules that were designed to deliver venom into a victim are lifted, sorted, and rerouted, diverted from their original purpose into a new architecture where they will be held in reserve. The attack has not failed. It has been appropriated. The sea slug does not merely survive the weapon, it converts the weapon into its own. Nudibranches, a diverse group of shell-less gastropods, operate at the boundary between vulnerability and dominance, their bodies lacking the rigid defenses that characterize many marine organisms, yet possessing a capacity to manipulate chemistry that rivals the most heavily armed species. Their strategy begins with feeding, but not in the conventional sense of simple consumption. When a nudibranch grazes on toxic sponges, cnidarians, or other chemically defended organisms, it is not just extracting nutrients, it is sampling and sequestering secondary metabolites, compounds evolved by the prey as deterrence. These molecules, often complex and energetically expensive to synthesize, are not broken down and discarded. Instead, they are transported through the digestive system and incorporated into the slug's own tissues, where they are stored in specialized glands or distributed across the body surface. This process requires a level of biochemical tolerance that is itself a significant adaptation. The toxins that the nudibranch consumes are designed to disrupt cellular function, targeting membranes, enzymes, or signaling pathways. To avoid self-harm, the slug must possess mechanisms that neutralize or compartmentalize these compounds during transport, preventing them from interacting with sensitive internal structures. This may involve binding proteins that sequester toxins, modifications to membrane composition that reduce susceptibility, or enzymatic pathways that alter the compounds just enough to render them safe for storage, while preserving their defensive properties. The result is a system where the line between digestion and defense becomes blurred, and where the act of feeding is inseparable from the construction of a chemical arsenal. In some species, the process extends beyond molecular sequestration into the realm of cellular theft. Nudibranchs that feed on cnidarians, organisms such as hydroids or jellyfish, are capable of isolating intact nematocysts from their prey and transporting them to the tips of their own appendages, known as serrata. These stolen stinging cells, called kleptoconidae, are not merely stored, they remain functional, ready to discharge when the slug is threatened. The Nudibranch effectively becomes a mobile extension of the cnidarian's defense system, deploying borrowed weapons with the same lethality as their original owner. This is not mimicry or imitation. It is direct incorporation, a form of biological recycling that repurposes entire cellular structures. The placement of these defenses is not arbitrary. Serrata, the finger-like projections that cover the bodies of many nudibranchs, serve as both storage sites and sacrificial structures. When a predator makes contact, it often targets these appendages first, encountering a concentrated array of toxins or stinging cells. The slug can autotomize or shed damaged serrata, sacrificing a portion of its body to escape while retaining the rest of its chemical defenses. This arrangement creates a layered system where the outermost structures absorb the initial impact, delivering a deterrent while protecting the core of the organism. The loss of serrata is not trivial, but it is a calculated trade-off that preserves the animal's overall integrity. The visual appearance of nudibranches reinforces their chemical strategy. Many species exhibit bright, contrasting colors: blues, oranges, yellows, and blacks arranged in patterns that stand out against the substrate. This aposematic coloration serves as a signal to potential predators, advertising the presence of chemical defenses. The effectiveness of this signal depends on the predator's ability to learn and remember associations between color and unpalatability or toxicity. Over time, individuals that attempt to consume a nudibranch and experience the consequences, whether through taste, irritation, or physiological effects, may avoid similarly colored organisms in the future. The coloration, therefore, extends the reach of the chemical defense beyond direct contact, influencing behavior through memory. Yet this apparent boldness introduces a reversal that complicates the narrative of vulnerability. The nudibranch, lacking a shell and moving slowly, would seem to be at a disadvantage in a predator-rich environment. Instead, its reliance on stolen chemistry allows it to occupy a niche where visibility becomes an asset rather than a liability. By advertising its defenses, the slug reduces the likelihood of attack, conserving energy that would otherwise be spent on escape or concealment. The absence of physical protection is compensated by a system that deters interaction before it begins, transforming exposure into a form of control. The ecological implications of this strategy are far-reaching. By feeding on toxic prey and incorporating their defenses, nudibranches influence the distribution and abundance of those prey species. At the same time, their own presence affects the behavior of predators, which must navigate a landscape where certain signals indicate high risk. This creates a network of interactions where chemical compounds flow through the ecosystem, transferred from one organism to another and shaping relationships at multiple levels. The nudibranch becomes a conduit in this network, linking the chemical ecology of its prey to the broader community. The diversity of compounds involved in this process is remarkable. Sponges, for example, produce a wide array of secondary metabolites with antibacterial, antifungal, or cytotoxic properties, while cnidarians rely on venomous proteins and peptides to capture prey and deter predators. Nudibranches that specialize on different prey types accumulate distinct chemical profiles, leading to variation in their defensive capabilities. This specialization can drive diversification within the group, as different species adapt to different chemical landscapes, each developing unique combinations of tolerance, sequestration, and display. At the physiological level, the integration of these compounds into the nudibranch's body requires precise regulation. Storage sites must isolate toxins from sensitive tissues, and mechanisms must exist to release or activate the compounds when needed. In the case of Kleptoconidae, the transport of intact nematocysts to the cerata involves cellular processes that maintain the integrity of these structures, ensuring that they remain functional despite being removed from their original context. This level of control suggests a complex interplay between the slug's own cells and the stolen components, a relationship that blurs the boundaries between organism and tool. The evolutionary pathway that leads to such a system is shaped by both opportunity and constraint. The availability of toxic prey provides the raw material for chemical acquisition, while the absence of a protective shell creates selective pressure for alternative defenses. Over time, individuals that can tolerate and utilize these compounds gain an advantage, leading to the refinement of the sequestration process. This co-evolutionary dynamic can also influence the prey species, which may alter their chemical profiles in response to predation, creating a feedback loop that drives further adaptation. In this context, the Nudibranch's strategy can be seen as a form of chemical parasitism, where the defenses of one organism are co-opted by another. However, it is also a form of innovation, transforming existing compounds and structures into new configurations that serve different purposes. The slug does not create its weapons from scratch. It assembles them from the environment, selecting, modifying, and deploying them in ways that reflect its own ecological needs. This approach reduces the metabolic burden of synthesizing complex toxins while allowing access to a wide range of defensive options. As the nudibranch continues its slow movement across the substrate, its cerata sway with the currents, each one a repository of borrowed chemistry. The reef around it remains active, filled with organisms that hunt, hide, and compete. Yet the slug occupies a distinct role within this system. It does not rely on speed or strength, nor does it produce its own arsenal in the traditional sense. Instead, it operates as a collector and redistributor of chemical defenses, a participant in a cycle where the boundaries between prey and predator, between weapon and resource, are constantly renegotiated. The chemical thief does not simply take, it transforms. By converting food into defense, by turning the act of consumption into an act of acquisition, the nudibranch exemplifies a strategy that is both efficient and profound. It reveals a dimension of survival where the most effective solutions are not always those that generate new capabilities, but those that repurpose existing ones, integrating them into systems that extend beyond the individual. In doing so, it reshapes the landscape of interaction, demonstrating that, in the ocean, power can be derived not only from what an organism produces, but from what it is able to claim and make its own. The inhale begins before the prey registers a predator exists. A filament twitches. No, not a filament, a lure, and a curious fish drifts closer to inspect what appears to be a harmless scrap of drifting life. And in that instant, the water itself collapses inward. There is no chase, no acceleration, only a sudden absence as the space in front of the fish becomes a vacuum and the fish becomes part of it. The predator does not lunge forward. It pulls the world toward itself, and the encounter ends before it can be recognized as one. What looked like rock has fed, and the reef returns to stillness as if nothing has occurred. The frogfish does not operate through venom, yet its strategy aligns with the same governing principle as chemical warfare: control perception, control outcome. Its body is a mutable interface between organism and environment, a surface engineered to dissolve into the visual and chemical noise of the reef. The skin is covered in dermal appendages, irregular projections that mimic algae, sponge tissue, or coral polyps, while chromatophores adjust coloration to match the surrounding substrate with remarkable precision. This is not generic camouflage. It is contextual mimicry, tuned to specific microhabitats, where the difference between survival and failure is measured in how convincingly an organism can become something it is not. The illusion extends beyond appearance into behavior. Frogfish remain almost perfectly motionless, a stillness that is itself a signal within the reef, where constant movement is the norm. By not moving, the frogfish aligns itself with the category of objects that are ignored: rock, coral, debris, exploiting the filtering mechanisms of prey species that must prioritize certain stimuli over others. This behavioral mimicry reduces the likelihood of detection not by hiding completely, but by being classified incorrectly by the sensory systems of other organisms. The frogfish is seen, but it is not recognized. This misclassification is reinforced by chemical subtlety. While not producing toxins or deterrent compounds, the frogfish's skin can harbor microbial communities and surface chemistries that blend with the surrounding environment, reducing the emission of distinctive cues that might otherwise betray its presence. In aquatic systems, where chemical gradients provide critical information about food, predators, and habitat, minimizing or masking these signals contributes to the overall illusion. The frogfish does not need to emit a strong chemical signature. It benefits from becoming chemically unremarkable, a background rather than a signal. The hunting mechanism that completes this system is based on speed and precision rather than force. The alecium, a modified dorsal spine, extends forward and ends in a fleshy lure known as the esca. This structure is often shaped and colored to resemble prey items such as small fish, worms, or crustaceans, and it can be moved independently of the body to simulate life. The lure does not simply attract attention, it creates a focal point that draws the prey into a specific spatial relationship with the frogfish's mouth. This positioning is critical because the strike itself is not a forward motion, but an inward collapse. When the moment arrives, the frogfish expands its buccal cavity with extraordinary rapidity, generating a suction force that pulls water and anything within it into its mouth. The speed of this expansion is such that the prey is captured before it can initiate an escape response. This is not a contest of reaction times. It is a preemption of reaction entirely. The prey's sensory systems, already misled by the illusion, are further overwhelmed by the suddenness of the event, leaving no window for correction. The frogfish does not outpace its prey. It removes the need for pursuit by ensuring that the prey is already within range when the strike occurs. The efficiency of this system reflects a deep integration of form, behavior, and environment. By minimizing movement and maximizing disguise, the frogfish reduces the energetic cost associated with active hunting. It does not need to patrol territory or engage in prolonged chases. It waits, allowing the dynamics of the reef to bring opportunities within reach. This strategy is particularly effective in habitats where prey density is high and movement patterns are predictable, such as coral reefs and sponge-dominated environments. The frogfish becomes a fixed point within a flow of organisms, a node where interactions are resolved quickly and decisively. A reversal emerges when considering the frogfish's apparent limitations. Its body is not built for sustained swimming. Its movements are slow and deliberate, and it lacks the defensive toxins that characterize many other marine species. Yet these constraints are precisely what enable its success. The inability to chase drives the evolution of a system that eliminates the need to chase, while the absence of chemical defenses is compensated by a level of concealment that reduces the likelihood of being targeted. The frogfish does not need to deter predators chemically if it can avoid being identified as prey or predator altogether. This avoidance is not absolute. Certain predators, particularly those that rely on broader sensory cues or that have learned to recognize frogfish, can still pose a threat. In these situations, the frogfish may rely on secondary strategies such as sudden movements, changes in coloration, or the use of its pectoral fins to walk along the substrate to a new location. These movements are limited in scope, but sufficient to reposition the animal within a more favorable context. Again, the emphasis is on altering perception and position rather than engaging directly. At the ecological level, the frogfish's role is subtle but significant. By preying on small fish and invertebrates, it contributes to the regulation of these populations, influencing the balance of species within its habitat. Its presence also affects the behavior of prey species, which may alter their movement patterns or avoid certain areas based on past encounters or learned associations. This creates a feedback loop where the frogfish's hunting strategy shapes the distribution and behavior of other organisms, reinforcing its effectiveness over time. The concept of chemical illusion in this context extends beyond the absence of toxins to the manipulation of sensory expectations. The frogfish does not produce compounds that incapacitate or deter. It operates within the chemical landscape of the reef, aligning itself with the background and avoiding the creation of signals that would trigger attention. This alignment is a form of environmental integration, where the organism becomes part of the system it inhabits, indistinguishable from the elements that surround it. The illusion is maintained not through active suppression of detection, but through the absence of distinguishing features. The evolutionary pathway that leads to this strategy involves a series of trade-offs and refinements. The development of specialized skin structures, the reduction of locomotor capability, and the enhancement of suction feeding all contribute to a system optimized for ambush. Each adaptation reinforces the others, creating a cohesive strategy that is greater than the sum of its parts. The frogfish does not excel in any single domain when considered in isolation, but its integration of multiple modest capabilities results in a highly effective mode of predation. As the captured prey disappears into the frogfish's mouth, the surrounding environment resumes its apparent normality. Other fish continue to move through the water, currents shift, and light patterns change across the reef. The frogfish remains, indistinguishable from the substrate, its presence concealed not by absence, but by assimilation. The interaction leaves no visible trace, no disturbance that signals what has occurred, yet it represents a precise intervention in the flow of life within the ecosystem. In this way, the frogfish exemplifies a form of warfare that operates without toxins, without overt displays of, and without sustained engagement. It is a strategy built on the control of perception, where the outcome is determined not by the ability to overpower, but by the ability to remain unrecognized until the moment of action. The illusion hunter does not need to convince its prey of danger. It needs to convince it of safety, creating a context in which the prey's own expectations lead it into the trap. The reef, dense with signals and interactions, becomes a medium through which the frogfish exerts its influence. by aligning itself with the patterns and cues that define the environment, it transforms from an organism into a condition, a possibility embedded within the fabric of the habitat. The prey does not encounter the frogfish as a distinct entity. It encounters it as part of the world, and in doing so, it becomes part of the frogfish's strategy.